Introduce the session chair and the first speaker, who's Ben Neel. I've known Ben for quite a long time. Ben was has been or was associated with Harvard for a very long time. He was originally a postdoc in Ray Erickson's lab beginning in 1985. In 1988, he began to move up through the ranks of Harvard Medical School, beginning as assistant professor and then culminating in professor of medicine in 1999. He was the director of the cancer biology program at the Beth Israel Hospital, which then became the Beth Israel Deaconess Medical Center since 1994, and the deputy director of basic research in the hematology division there as well. In 2006, he became the William B. Castle Chair of Medicine in HMS, at HMS, and then in 2007, he migrated northward to become the director of the Ontario Cancer Institute and joined the faculty at the University of Toronto. It's a great pleasure to welcome Ben back.
So I'd like to start out by thanking Mike and Forrest for inviting me, and Cell Signaling for sponsoring such a great meeting, and all of you for staying through this long but nevertheless exciting day. So what I thought I'd talk about today is some recent work that's a little different than what you've heard about so far, which is a role for signaling proteins in the control of cell polarity. And most of what I'm going to talk about in the first part of the talk has just been published, so I'm going to try to go through that reasonably quickly and to get to some new unpublished data at the end. But although that's the official title of my talk, it hasn't escaped my attention that as an American expat going to Toronto, it's a little bit ironic to be speaking on Election Day in the Koch Institute. So I'm going to come back to this theme also right at the end of my talk, so stay tuned. So just so everybody has a brief introduction to what I'm going to talk about, our lab has been interested in tyrosine phosphatases for a long time, and I'll have a little bit to say about the tyrosine phosphatase SHP2 at the end of the talk. But the reason we became interested in scaffolding adapters like Gab1, which you heard about briefly from Luke Cantley earlier in the day, was because it's an SHP2 binding protein. And Gab1 is a scaffolding protein that, in addition to activating SHP2, also activates the PI3 kinase pathway. It's a critical adapter in multiple RTK and cytokine signaling systems. It's probably best known for its role in HGF receptor signaling, where it promotes epithelial cell scattering and morphogenesis. And as I already alluded to, it binds SHP2 and activates ERK, and it binds P85 and activates PI3 kinase and also has some other effects. And it's part of a small scaffolding adapter family that includes Gab2 and Gab3, and I'll come back to Gab2 at the end of the talk. Now, this is a very simplified cartoon, much more simple than you've seen from some of my colleagues earlier, like Doug Laufenberger, about how Gab proteins are believed to promote cell scattering downstream of HGF. So when HGF binds to its receptor, it recruits Gab1 to the receptor. Gab1 becomes tyrosine phosphorylated, binds SHP2, and that activates RAS and Rho, and that leads to cell scattering and cell proliferation. Now, interestingly, bacteria, in particular H. pylori, have co-opted this program in the form of a virulence determinant called CAG-A. And CAG-A is an essential virulence determinant in H. pylori, which is, for those of you who don't know, is the major cause of both gastric ulcer and gastric carcinoma worldwide. And CAG-A is basically like a mini Gab protein. It's injected by the H. pylori into gastric epithelial cells, where it becomes tyrosine phosphorylated by Src family kinases, binds to SHP2, and does the same thing as Gab proteins. It activates the RAS-ERK pathway and activates Rho. And it produces what Masanori Hatakeyama, whose lab has done the vast majority of the work in this area, calls the hummingbird phenotype, but I think anyone looking at these cells would say it's cell scattering, and they also cause cell proliferation and depending on the context, apoptosis. Now, a couple of years ago, actually right around the time I moved to Toronto, I didn't have anybody in my lab, so I had a lot of time to read. Now I have too many people in my lab and have no time to read. So I actually came across this paper that had just been published by Hatakeyama's lab that reported another function for CAG-A, and that is that CAG-A bound to the polarity kinase PAR1 through the kinase domain and inhibited PAR1 kinase activity. And through functional studies, Hatakeyama's lab found that CAG-A disrupted cell polarity, and found that both SHP2 binding sites in CAG-A and the PAR1 interaction domain was required for CAG-A to promote cell scattering. So before going any further, let me just make sure everyone knows what PAR proteins are. So PAR proteins get their name from work in C. elegans, largely by Ken Kempfes. And these are essential regulators of epithelial cell polarity. And there are several members of the PAR protein family, including PAR1 and PAR4, which are protein kinases. You've already heard about PAR4 in this meeting by its better-known name, which is LKB1. So PAR4 is LKB1. PAR3 and PAR6 are scaffold proteins. PAR5 is a 14-3-3 ortholog. And PAR2 is the only member that is not known, or at least not known to me, to be conserved in mammalian cells, and it's a ring domain protein. Now, the PAR proteins form multi-protein complexes, and they're essential regulators of epithelial cell polarity. For example, the PAR3, PAR6 APKC complex, which is held together by the interactions that are diagrammed on this cartoon, is localized at and essential for the formation of tight junctions in polarized epithelial cells. And some of the evidence for this is that PAR3 depletion delays tight junction formation in so-called calcium switch assays, which I'll show you a little bit later in the talk. And APKC depleted cells fail to form tight junctions. Now, the PAR3, PAR6 APKC complex engages in a mutually exclusive interaction with PAR1 and the 14-3-3 or PAR5 complex. So PAR1 and the PAR3, PAR6 APKC complex are mutually exclusive for localization, and that's because PAR1 is located not in the tight junctions, but in the lateral domain. The evidence is that PAR1 and PAR6 and the PAR3, PAR6 APKC complex are also functionally exclusive. And the details by which PAR1 regulates the PAR3, PAR6 APKC complex have not been clear, and that's what I'm going to hopefully clarify, at least from our view, in the first part of the talk. So again, some of the evidence for this is that PAR1 depletion causes shortening of the lateral domain of polarized epithelial cells and increases transepithelial resistance, which is a measure of tight junction formation and integrity. So based on that work by Hatakeyama, which showed that KAG-A blocked the polarity complexes, we wondered whether, because KAG-A and Gab1 have other similar functions, do Gab proteins like KAG-A bind to PAR1 to control cell polarity? And for those of you who want the take-home message, the answer is yes, but in our view, in an entirely different way. So what's the evidence for this? So all the work that I'm going to show you now has been done by a single, very assiduous and talented postdoc who's actually just going on the job market also, Zicheng Yang. And so first he wanted to know whether there was any evidence that Gab proteins could interact with PAR1. So he carried out transient co-transfection assays where he co-transfected either HA-Gab1 or HA-Gab2 with PAR1b. That was flag tag, and he found that in fact both of these Gab proteins could associate with PAR1b. And I'll just point out that actually Gab2 is expressed at much lower levels in this transient transfection system, and it binds actually the same amount of PAR1b. So it turns out that PAR1 actually binds better to Gab2, and I'll have a little bit to say about that right at the end of the talk. So without belaboring the biochemistry, he went on to map the interaction domain on Gab1 that binds PAR1b to amino acids 152 to 250 by deletion mutagenesis. And I should say that we've recently been able to narrow this to a 15 amino acid peptide for which we have a NMR structure, or actually shouldn't say we, Masa Mitsu and Mitsu Okura at our institute have an NMR structure that supports the biochemistry that I'm going to tell you in the next couple of slides. Now, everything up until this point could be the same story as Hatakeyama's lab for KAG-A, only with Gab proteins. But it turns out that everything else was different. So unlike KAG-A, which binds to the kinase domain of PAR1 and inhibits its kinase domain, Gab1 binds to the C-terminus of PAR1b, the so-called kinase-associated domain. And not only does Gab1 fail to bind to the same domain as KAG-A, it actually activates PAR1 through binding to its C-terminal K domain. And I don't have time to show you what the mechanism is for this, but both the paper that I alluded to earlier and the unpublished structural studies that I have would support a model in which binding to the C-terminal Ka domain disrupts a basal autoinhibitory interaction between the C-terminal K domain and the kinase domain. So by binding to the K domain, Gab1 actually activates PAR1 kinase activity. Well, having already found one difference from the published results of KAG-A and PAR1, we wanted to know if Gab1 also bound other members of the polarity machinery. So Zicheng carried out similar experiments where he now co-transfected either PAR3, PAR6, or APKC. And you can see that Gab1 binds strongly to PAR3. It also binds to APKC. We think this is largely because PAR3 is bound to APKC under these circumstances and binds very weakly, if at all, to PAR6. So I won't belabor the mapping studies that he did to map the par3 interaction, but basically the par3 interaction occurs between amino acids 301 to 340 of Gab1 and involves an interaction with the first PDZ domain of par3. So if you just look at this simplified linear cartoon, and unlike the engineers in the audience, linearity is about all I can handle, especially at this time of the day, you can see that one could imagine that you could form a termolecular complex between par1 and par3. And in this way, Gab1 could bring par1 together with par3 and in principle could effect that mutually exclusive interaction on the lateral domain that I told you didn't have a detailed mechanism. So the question we wanted to know was, does Gab1 promote par1, par3 interaction? So again, Zichang carried out co-transfection experiments, and the take-home message is again that if you co-transfect in Gab1, you can bring down more par1, par3 complex. So what's the purpose of this interaction? OK, well, it turned out that he noticed that by looking very carefully at the gels, that when he co-transfected in Gab1, par3, and wild-type par1b, there was less Gab1 par3 complex than if he just looked at Gab1 and par3 alone. So when wild-type par1 comes into the complex, it releases par3 from Gab1. And this is a kinase-dependent process because if he mutates the kinase domain of par1b, you stabilize this termolecular complex. And this is not just true, all of the other stuff I showed you is also not just true in transient transfected cells, but this in particular is not just true in transient transfected cells. If you knock down Gab1 in a cell or in several different cell types that display epithelial polarity, in this case MDCK2 cells, and carry out a calcium switch assay, you can see that there's a decrease in par3 phosphorylation in the Gab1 knockdown cells. So as I told you earlier, Gab1 binds to par3 via this 301 to 340 amino acids on Gab1, and it binds to the PDZ domain of par3. And that same domain of par3 is a major binding region for par3 to interact with par6. So that raises the question of, do par6 and Gab1 complete for par3 binding? And again, if in fact you co-transfect Gab1, par3, and par6, you get a decrease in the Gab1, par3 interaction. So what that means is that Gab1 can actually strip apart the par3, par6 APKC complex, strip the par6 off, and just bind par3 APKC, allowing par1 to phosphorylate par3 and release it from the complex. And this, again, is not just true in transient transfected cells, because if you look in MDCK2 cells that express Gab1 and you deplete Gab1, you get an increase in the par3, par6 APKC complex. So to summarize a lot of complicated biochemistry, that leads to the following provisional model, that Gab1 binds to par1 through its M terminus, and it binds to par3 through its C terminus, and it can also disrupt par3's interaction with par6, and that par1b can then phosphorylate par3 and release it from the lateral membrane. So acting as a scaffold, Gab1 binds par1 and par3 into a transient complex, facilitating the phosphorylation of par3 by par1. And then once phosphorylated, par3 is released from that complex, and as I already mentioned, par6 and Gab1 compete for binding. OK, does any of this have any biological consequences? So in order to test this, we first wanted to know, is Gab1 localized on the lateral membrane of polarized epithelial cells? And in fact, if you look at HA tag Gab1 that's expressed at wild-type levels, we just needed that for a good antibody, Gab1 is recruited to the lateral domain of polarized epithelial cells, excluded from the Z01 or tight junction binding domain in a calcium switch assay. And I should say that this is actually consistent with a long ago observation in the SHB2 field, which is DOS, which is the Drosophila ortholog of Gab1, was reported when it was first identified to be located on adherence junctions in Drosophila by Mike Simon's lab. OK, so what happens? I should have asked for the lights to go down. But what happens when you actually deplete Gab1 and look at tight junction formation? So this slide is actually a little bit hard to see. But if you look at a calcium switch assay and you look at the formation of tight junctions as monitored by Z01 staining, you can see that it takes about an hour before the tight junctions actually start to really completely surround the cells. But if you knock down Gab1, just as you have as the effects of par1 depletion, you get a markedly accelerated formation of tight junctions. Now, I generally don't like immunofluorescence studies because I think they're too prone to investigator bias. Fortunately, there's actually a measurement that one can make to assess tight junction integrity, and that's transepithelial resistance. So when you get polarized epithelial cells and you form tight junctions, you get a resistance across the membrane. And so we could measure transepithelial resistance in the Gab1 knockdown cells, and you'll see that you get an increased rate of acquisition of transepithelial resistance and a higher final level of transepithelial resistance in the Gab1 knockdown cells. Again, consistent with the effects of PAR1 knockdown, when you deplete Gab1, you get shortening of the lateral membrane as measured by EEC adherent thickness. And these are the only experiments that were not done by Zi Cheng. They were done by Bin Wei, a graduate student in Senpil Muthuswami's lab. And you also get an increase in the percentage of the membrane that's occupied by Z01 or by tight junctions. Again, a morphological correlate of the increase in transepithelial resistance that we observed in the measurement studies. Okay, well, that says that Gab1 is required for determining the rate and extent of tight junction formation. Does this have anything to do with Gab1's ability to bind any of these polarity proteins? So we found that if you now look in Gab1 knockdown cells and then you re-express an RNAi-resistant form of Gab1, you can see that, again, you have decreased tight junction formation at 1 hour following a calcium switch. But if you do exactly the same experiment with either the Gab1 mutant that can't bind PAR1B, that mutant for that relatively big deletion mutant, or with the Gab1 mutant that can't bind PAR3, you don't have any effect on the accelerated tight junction formation. Now, the deletion the deletion that disrupts PAR1B binding, it's pretty big. It's 100 amino acids. And actually, it overlaps with a binding site for PAC4 that had been reported earlier by Morag Park's lab. So we were concerned about whether or not this was really going through PAR1B. So to address that, we did 2 types of experiments which led to the same result. The first one was we actually directly fused PAR1B to the C-terminus of the deletion mutant that was lacking the PAR1B binding site. And even though this mutant is expressed at very low levels, you get a really dramatic decrease in tight junction formation. Now, I didn't believe that either because it was really expressed at very low levels. So I thought it was some real artifact. So we actually made, we being Zi Cheng, made an inducible form of this same construct where he introduced an FKB PTAL to the deletion mutant of Gab1 and an FRB binding site to the PAR1B construct. And then in the presence of a rapamycin analog, you can induce the joining of these 2 constructs. And when you do that, again, you disrupt tight junction formation in the calcium switch assay. And you also have exactly the converse effect as Gab1 deletion on transepithelial resistance. So you can see that you decrease both the rate and the extent of transepithelial resistance acquisition after a calcium switch assay. And in addition, again, Gab1 is best known for its effects on doing the opposite of forming tight junctions, which is to promote cell scattering. So we carried out the same types of experiments and looked at the effects of Gab1 in these mutants on cell scattering. You can see that if you overexpress Gab1, you get a little loosening of the colonies to start with, and then you potentiate scattering in response to limiting doses of FGF. And this is blocked by the Gab1 mutant that binds PAR1, also blocked by the Gab1 mutant that doesn't bind PAR3. And it's really potentiated in this dimerizing system when we induce the formation of the Gab1 PAR complex. So to summarize what I've shown you so far, that what we think happens under the steady state conditions is that Gab1 is located on the lateral domain capable of binding PAR1. If a little PAR3 leaks off the lateral domain, it comes in contact with Gab1, is captured, phosphorylated by PAR1, comes off, and is able to be sequestered by the 14-3 ortholog in the cytoplasm. Now, in the case of tight junction formation in a calcium switch assay, it's sort of the converse. Calcium actually triggers the relocalization of Gab1 from the cytoplasm to the lateral membrane. And again, as in this region where Gab1 is bound, if any PAR3, PAR6 comes in, Gab1 first strips PAR6 away from PAR3, and then it phosphorylates PAR3. PAR1 phosphorylates PAR3, releasing it again to the cytoplasm and limiting the domain over which tight junctions can occur. So I just want to close with some new data, which has to do with is SHP2, which is the reason we were interested in this at all in the first place, also involved in regulating cell polarity. So again, Zicheng did the same type of experiments now with point mutants that disrupt the ability of SHP2 to bind to Gab1. And what you can see that, again, if you knock down with Gab1, you accelerate tight junction formation. If you knock down if you knock down and then restore wild-type Gab1, you disrupt tight junctions. If you restore it with a mutant that can't bind SHP2, the rate of acquisition of tight junctions is not impaired. And if you look at transepithelial resistance, what you see is that SHP2 is particularly important for the initial rate of tight junction acquisition, but actually it's able to be corrected by the presumably by the PAR3, PAR6 interactions that I showed you earlier. So we were wondering what the mechanism might be for SHP2 to regulate the PAR3, PAR6 APKC complex, and we noticed that in many of the reported databases, including phosphocyte, that there are peptides for PAR3 that's tyrosine phosphorylated and haven't really been studied that extensively. So we noticed that in a calcium switch assay that there's actually an increase in tyrosine phosphorylation of PAR3, and that this is accentuated in a Gab1 mutant that fails to bind SHP2. And this prompted us to look back in the literature, and there's actually one study that, from this group here that shows that, that purports that SART family kinases phosphorylate PAR3 on tyrosine residues, and that PAR3 then sequesters limb kinase and leads to tight junction stabilization by the model that I show on this slide. And so we just have some preliminary data that suggests that this may be at least part of the story, because if you look in the calcium switch assays of wild-type Gab1 reconstituted cells and SHP2 mutant reconstituted cells, which have increased PAR3 tyrosine phosphorylation, you can see that there's increased cofilin phosphorylation during this process, consistent with stabilization of tight junctions. So to summarize what I've told you today, we found that in addition to their role in regulating activation of ERK and AKT through SHP2 and P85 respectively, that Gab1 in particular is a scaffold for PAR1 and PAR3, and that by binding PAR1 and PAR3 in the same transient complex, Gab1 promotes PAR3 serine phosphorylation by PAR1 and simultaneously can strip PAR3 and PAR6 from each other and inhibit PAR3, PAR6 APKC complex formation. We've also recently found that Gab1, by binding SHP2, can contribute to the early regulation of tight junction formation after a calcium switch by controlling the extent of PAR3 tyrosine phosphorylation. And we believe that together these complexes are important for regulating the rate and degree to which tight junctions form and transepithelial resistance develops after calcium switch assays, and hopefully we'll be able to find similar effects in real mice. And I also showed you that the Gab1 PAR complex regulates cell scattering. So I just want to close, since this is a session on cancer. Obviously, polarity disruption is important in cancer, but there may be even more direct implication of this work that has to do with the point that I made on the earlier slide on the Gab-PAR complexes, which is that Gab2 is an oncogene, as was first shown by Momo Benetirizales in my lab, for breast cancer, and has subsequently been found in other cancers, including serous ovarian cancer and also possibly melanoma. And as we and Jim Griffin and Rick Van Etten showed, inappropriate phosphorylation of Gab2 is also critical for the transforming activity of BCR-ABL. So our data suggests that Gab2 PAR interactions might contribute to this oncogenic activity. And I'll just leave you with one interesting preliminary result. It's been repeated a couple times, but a provocative result which suggests this may be true. And that is that if you co-express Gab2 with ERB2 in MCF10A cells, you can promote a mesenchymal-like morphology on the MCF10A cells. But if you co-express a Gab2 mutant that's lacking the binding site that we've defined for Gab2, you don't cause this mesenchymal phenotype. So I want to stop here and thank the people who did this work. As I said, almost every experiment I showed you was done by Zicheng Yang. I mentioned the structural studies that are as yet unpublished from Mahasa and Mitsu, and the experiment that was done by Bin and in Senthal's lab. And these investigators helped us a lot with reagents and explaining cell polarity to me. And I just want to have one last slide coming back to the theme I mentioned earlier, which is if Romney wins, there's always Canada. This is my lab right here. And I have many positions available. Just give me a call. So, thanks.
Time for a question or two for Ben.
John.
Ben, can you distinguish effects on adherent junctions and tight junctions? Because it looked to me like there was also a weakening of cell-cell adhesion. I mean, adherent junctions.
Well, there's definitely weakening of cell-cell adhesion when you stimulate with HGF. And remember, those weren't done serum-free, so those may be serum effects. It's possible. I mean, one of the problems is that adherence junctions are required to set up tight junctions. So we can't really exclude that possibility, but given the effects on the localization of the PAR, I would say that, for example, the PAR3 APKC complex localization goes in the same direction you would expect from Z01. So we still can't exclude an effect on the adherence junctions.
If I understand your data correctly, and I may have misunderstood it, most of the data you showed had to do with tight junction formation, and then you showed some stuff about scattering. But I think this is what Joan's question was getting at as well, is do you think that the same process is involved in the disassembly?
Right.
So that's what you're saying, but you don't have clear data that phosphorylation event and all happens during, you showed that it's involved in scattering.
Right,
But
and the sites that
the
bind
sites
to
are
those
important because the
proteins
mutants
are
don't do
critical.
it.
And it can be rescued by the direct fusion.
Okay.
So yeah, I mean, it's harder to see. The sites aren't that great. The antibodies aren't that great for this area, so maybe cell signaling can make some new ones.
There you go.
But,
Okay, if not, let's thank Ben one more time.
okay.